Hello students. In the previous session, we had studied about the characteristics of the genetic material DNA and RNA, and we had discussed about the stability, about the replicability, about the even about the mutatability, and even the expressibility of genetic material. With respect to four characteristics, we had seen which is the most abundant genetic material, that is DNA, and also we had studied the reason behind that while studying about those characteristics. And also we had studied about the RNA world, how the RNA was the predominant genetic material once upon a time, and how it was later replaced by the DNA molecule, and why it was done so, all that. And in today's session, let us consider the replication, the process mechanism of replication of DNA. And in the previous session itself, I had told you replication is the process of production of a replica of the DNA molecule, and this takes place in the S phase of the cell cycle. It takes place in the S phase of cell cycle. See, I had told you while teaching you about the cell cycle and cell division in the previous year that you need to understand that aspect completely and clearly, because see, only when you understand that aspect you will be able to link that to here. Okay, so S phase is also called as synthetic phase, where DNA, if it is amount of the DNA before the S phase is 2C, after the S phase will become doubled, that is 4C. How does it happen? How is it going to happen? It is because of the process and the mechanism of replication, and this mechanism of replication was also proposed immediately after the proposition of the double helix structure of the DNA in the year 1953 by James Watson and Francis Crick in the year 1953. So these people who were awarded the Nobel Prize for the discovery, for the proposition of the DNA double helix, they said that it came to our notice that means we did not miss to see that the base pairing of the sequences or the nitrogen bases in the double helix structure that we have proposed is also responsible for what? Replicability of the DNA. And hence they also went on to propose the mechanism of replication and the enzyme machinery of the replication also. And the replication mechanism that was given by Watson and Crick in the year 1953 was called as the semi-conservative replication. Semi-conservative replication. What is the meaning of this semi-conservative replication? You should know, you know the meaning of conservation. Conservation means saving a part of it. Okay, saving. Conservation means just saving. Semi-conservative replication refers to, see, if this is the DNA structure, if this is the molecule of the DNA, during the replication what happens here is that one strand will be retained in the newly produced two DNA strands or two DNA molecules. See, this will open up like this, and based on the complementarity base pairing, new strand is produced on this one. This will act as the guiding strand, also called as the complementary strand on the Karitaveli. Okay, the older DNA will be having two strands, right? So both the strands will act to guide the synthesis of the new DNA strand here. So the new DNA strand or the nascent DNA strand is being produced completely and purely based on the complementarity base pairs, right? So two new DNA strands are being produced, right? And the two new DNA strands which are being produced, they of course are complementary to each other, but they both of them will not come together to form one new DNA molecule. According to this theory, what happens? See, entire molecule of the DNA is replicated, and then see, after the replication, the new DNA, replicated DNA, will look somewhat like this. I hope you can differentiate the colors. So one strand will be of the parent strand. One new strand that is produced is nothing but that of the newly synthesized strand. Okay, the saffron colored strand what you are seeing, it is a new strand, and the yellow strand what you are seeing, it is the old strand. That means a part of the parent DNA. This is the parent DNA, right? So the daughter DNA is being produced by the parent DNA based on the complementarity based pairing. But still, a part of the parent DNA, one strand of the parent DNA also comes along with a strand of the newly synthesized DNA. And this partly remaining DNA strand, and this mechanism, that is why it is called as semi-conservative replication of mechanism. Okay, so this semi-conservative replication of mechanism, of course there are different mechanisms of replication. Conservative theory of replication is there, okay, and there are other theories also, but for our syllabus we are studying the semi-conservative replication of the mechanism. Remember who proposed it? It can be a 1 mark important question. It was proposed by James Watson and Francis Crick in the year 1953, immediately after the proposition of double helix structure of the DNA. So I would read out the statement given by these two people. Okay, I'll just read out the statement for you. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. See, this was the statement that was given by Watson and Crick in order to showcase that themselves they actually postulated the mechanism of replication also. Now let us study the replication of DNA. We are studying about the process of replication. I mean about the replication of the DNA. We have not yet started the mechanism of replication. Before we go and understand the mechanism of replication, we need to study the experimental proof that was given to show that replication occurs by semi-conservative method. See, the replication mechanism that was explained by Watson and Crick was a semi-conservative replication mechanism that we already know, right? But this was experimentally proved by two people by name Michelson and Stahl. Okay, that is Matthew Michelson and Franklin Stahl. The complete name of these people, Matthew Michelson. You need to remember the name of the scientist, and Franklin Stahl. Okay, so these two people in the year 1958, they conducted an experiment. They conducted an experiment to show that DNA replication is occurring in what way? That is semi-conservative way. How is it possible and what experiment was done? See, this experiment is popularly known as Michelson-Stahl experiment, or in the examination they may ask you to write what experimental proof for semi-conservative replication. Okay, both ways it can be named. Now let us see what experiment was conducted by these two people. See, they considered the E. coli bacteria. Okay, so it is the favorite of all people, E. coli bacteria, and they had considered the DNA from E. coli bacteria. That is because E. coli, Escherichia coli is the simplest prokaryotic system with which the system of replication or the mechanism of replication could be easily understood. So what did they do here is they isolated the DNA of the E. coli, and they also took two isotopes of the nitrogen. You know, the atomic mass of nitrogen is 14, right? N14. But they considered a heavy isotope of nitrogen that was N15. Right? What experiment was conducted here by these two people is that they considered E. coli in two cultures. There was one Petri plate in which bacteria were cultured for many generations on the normal, that is N14 medium. That means, see, when culturing takes place, these E. coli, they are going to multiply, or one life cycle of E. coli will be completed in 20 minutes. ಅಂದ್ರೆ 20 ನಿಮಿಷಕ್ಕೆ one life cycle ಮುಗಿದಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ they are going to reproduce, they are going to produce the next generation, and the older generation will die, right? And if at all the next generation has to be produced, the DNA replication has to happen. Replication ಆಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ it has to utilize the nutritional medium appropriately in order to produce the new strand of the DNA. And what all the nutrition is required to produce the DNA? You already know the constituent of the DNA. The DNA will be having the bases. Nitrogen bases will be there, the pentose sugars will be there, the phosphate groups will be there, hydrogen bonds will be there, yes or no? In order to make all these things, the nutritional medium should be consisting of all these sources. So the nitrogen source must be there in order to replicate, right? In order to continue the life cycle of these bacteria, right? When they cultured on the N14 medium for many generations, what did they do here was they had provided all the necessary nutrients and normal nitrogen was given here. Yes, normal nitrogen was given and with all other nutrients, right? And after growing the bacteria for many generations, they transferred this batch of bacteria to a Petri plate that was supplemented with N15 nitrogen. Andre illi N14 normal nitrogen ittu, whereas in this case the heavy nitrogen was there. The only source of nitrogen in this case was what? Heavy nitrogen. That means these bacteria which were all having the normal nitrogen, when they were transferred to the new medium consisting of the heavy nitrogen, they have no other option than using that source of heavy nitrogen for the production of new DNA strand, right? See, on this new medium, they were allowed to germinate, they were allowed to not germinate, they were allowed to replicate and multiply and produce only one generation. That is only for 20 minutes they were cultured on this new medium. ಎಲ್ಲಿಂದ ಎಲ್ಲಿಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಿದ್ದಾರೆ? N14 ಇಂದ N15 ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಿದ್ದಾರೆ and they were made to grow, they were allowed to grow only for 20 minutes. That means that only one generation of the bacteria was produced. And now this newly produced bacteria is having what? It is having N15 DNA. Let me see, the DNA present in this was the N14. I am writing it with the saffron colored chalk here, okay? Whereas in this case, what happened? Replication took place, right? Because of this replication, semi-conservative replication took place, right? See what happened. This while this was replicated, it was opened up like this, right? So the new DNA that was produced was N15 DNA. I am showing it to the green chalk. N15. This is N14. Yes or no? Now, this newly produced molecule of the DNA will be consisting of half N14 and half N15. Yes or no? Half N14 and half N15 DNA is present in the newly synthesized DNA strand. So this confirms that half of the parent DNA is retained in the next generation, and that is how it is what? Semi-conservative replication. But how was this proved? How was this visualized by this experiment? They considered E. coli bacteria, they considered the heavy nitrogen source, right? Nutrient medium of the heavy nitrogen source. But along with that, they also performed one more technique called gradient centrifugation technique. That was called as gradient centrifugation technique. Gradient centrifugation technique by the usage of a chemical called cesium chloride. CSCl stands for cesium chloride. Why particularly this particular chemical was used in this experiment was, here they understood that this CSCl has a more density. It is much more heavier than the DNA. The density of the CSCl is much more than that of the DNA, and obviously the DNA molecule will float in the CSCl solution. That is the reason why it was used. Now what did they do was, see, when they had put this DNA, that means when they had taken the solution of the extracted DNA, so they had noted the position of the N14 DNA. N14 DNA, they had noted the position, right? And they had also marked the position of the N15 DNA separately, and when they transferred this culture or this batch of bacteria from N14 source to N15 source, N14 in the N15 ಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಿದಾಗ they thought that it is a completely a new source of DNA, because it can be now called as heavy DNA, because it is having the heavy isotope of the nitrogen, heavy DNA irutte, and that is why they thought that, see, in this test tube they could get what, N14 above and N15 below, because it is heavier DNA. That is what the assumption that they had made. But to their wonder, what happened here was, in the test tube they got to see both N14 and N15 forming a band together. Only one band was formed combined together, that is N14 and N15. Okay, so this consisted of N14 and N15. So this is how, with the help of gradient centrifugation technique, with the help of CSCl, cesium chloride solution, they once again proved that semi-conservative replication takes place in the prokaryotic and eukaryotic cell. That means a replication mechanism is taking place with the help of which mechanism? Semi-conservative mechanism. See, this experiment is a very important experiment for 5 marks. Diagram is also mentioned in the textbook. Simple experiment is there, simple diagrams are there. Okay, so you need to practice the diagram, and you learn this experiment, understand this experiment, and you should know how to write and explain for 5 marks. Okay, in the next session we are going to discuss about the mechanism and enzyme machinery of replication process. Thank you.